Welcome to the Chalkless class. This lesson is about human development from a single-cell zygote to its fixation in the wall of the mother's womb.
Human pregnancy begins with the fusion of male and female gametes in the female reproductive tract. The fertilized egg undergoes a series of mitotic divisions. Now it is called an embryo. Morula, blastocyst, and gastrula are the embryonic stages that appear during development. At the morula stage, the embryo enters the uterus and develops into a blastocyst. The blastocyst sinks into the uterine lining to get nourishment from the maternal side. This process is called implantation. The implanted embryo undergoes many changes and acquires the germinal layers, in a process called gastrulation. From these germinal layers various types of organs are formed. This process is called organogenesis. The embryo is protected, nourished, and respired in the womb of the mother, with the help of fetal membranes. The fetal membranes have an extraembryonic origin. By the end of 9th month of pregnancy, the fetus is fully developed and ready for delivery. The ovulated egg is a secondary oocyte. It is surrounded inside to outside by zona pellucida, corona radiata, and outer granulosa cells. During copulation, semen is released into the female genital tract. This process is known as insemination. The ejaculated sperm can survive 48 to 72 hours. The fertilization can occur if the ovum and sperms are transported simultaneously to the ampullary isthmic junction. That's why all copulations do not lead to fertilization and pregnancy. The process of fusion of male and female gametes is called fertilization. Only one sperm is allowed to enter the unfertilized egg. If the egg failed to fuse with the male gamete, it cannot survive in the female genital tract. It degenerates and is phagocytized. After fertilization, the fertilized egg is prevented from disintegration within the female reproductive tract. It begins cleavage that continues for several days. During this period, zygote is covered by zona pellucida. The corona radiata is lost within 2 days of the beginning of the cleavage. Zona pellucida prevents any additional spermatozoa from entering the zygote. It prevents premature implantation of the embryo into the wall of the fallopian tube. First cleavage occurs in a meridian plane after 30 hours of fertilization. The cleavage division completely divides the zygote, and the formed cells are almost equal in size. Such type of cleavage is called holoblastic and equal cleavage. The formed cells are blastomeres. The second cleavage also occurs in the meridional plane, at right angles to first cleavage, after 60 hours of fertilization. After the 2-cell stage, cleavage is asynchronous. In asynchronous cleavage, all the blastomeres do not divide simultaneously. One of the 2 blastomeres divides first into a meridional plane and produces 2 daughter cells. The cleavage division is delayed in another blastomere. At this stage, the embryo consists of 3 cells. It is a transient stage between 2 and 4 cell stages. A 4-cell stage is formed after the completion of the second cleavage in both blastomeres. The third cleavage is horizontal. It is slightly unequal. It forms 8 blastomeres. After third cleavage, the cells are held together by tight junctions. After the third cleavage, the rate and patterns of cleavage are nonspecific. In mammals, cleavage divisions are among the slowest in the animal kingdom. After the 8-cell stage, the embryo enters a phase called compaction. At this phase, the outer blastomeres tightly adhere by gap and tight junctions, and lose their individual identity when viewed from the surface. The outer cells form trophoectoderm and inner blastomeres produce inner cell mass. The peripheral cell layer secretes a fluid into the interior, creating a cavity called blastocyst. This process, which occurs about 4 days of fertilization, is called cavitation. The inner cells are pushed to a side as embryonal knob by the blastocyst. With the formation of blastocyst, the morula transforms into a stage called a blastocyst. A blastocyst has an outer cell wall trophoblast and a small group of blastomeres, the inner cell mass or embryonal knob. The inner cell mass gives rise to the body of the embryo and extraembryonic membranes, while the trophoblast forms only extraembryonic structures. At the blastocyst stage, the embryo is still surrounded by the zona pellucida. To facilitate the implantation, the blastocyst is released from zona pellucida in a process called hatching. A small region of the zona pellucida, usually directly covering the inner cell mass, dissolves, and the blastocyst emerges out from a hole into the uterine cavity. A protease enzyme causes the disintegration of zona pellucida. At the time of hatching, if the blastocyst breaks into 2 parts, both parts have a chance to develop into identical twins. Just before the embryo implants into the uterine wall, a significant change occurs in inner cell mass. A thin layer of cells appears ventral to the inner cell mass. At this stage, the embryonic disc contains a lower layer of hypoblast and the main upper layer of cells called epiblast. The hypoblast or primitive endoderm lines the yolk sac cavity. The epiblast contains the cells that make up the embryo itself, but extraembryonic tissues also arise. In the early phase of implantation, the blastocyst penetrates the uterine epithelium. The differentiation of cellular trophoblast occurs just before its implantation. The cellular trophoblast divides into outer syncytiotrophoblast and inner cytotrophoblast. Small projections of syncytiotrophoblast insert themselves between uterine epithelial cells. The entire blastocyst is enclosed in the endometrium. The next layer that appears after the hypoblast is the amnion. It is a layer of extraembryonic ectoderm that completely encloses the embryo. About 9 days after fertilization, hypoblast begins to spread and lines the cytotrophoblast, known as the extraembryonic endoderm or parietal layer. Cytotrophoblast forms primary trophoblastic villi which penetrate the endometrium. The posterior cells of the epiblast roll in and form an extraembryonic mesoderm between the yolk sac and trophoblast. After the implantation, the endometrial cells accumulate glycogen and lipid droplets. Now the endometrium is termed decidua. Decidua that overlies the embryo and its chorionic villi is the decidua capsularis. The decidua present between the embryo and myometrium of the uterine wall is decidua basalis. The remaining endometrium, which consists of decidual endometrial tissue, is called decidua parietalis.
Thank you for visiting the Chocklist class.